Hello, guys. Hope you can hear me. Hello everyone. My name is Shomo. Mm, I am the PMRFTA for this course, Computational Systems Biology, offered by NPTEL. I will be taking 2 hour live sessions each week on Tuesdays, 6 to 8 p.m. So please join the learners who have enrolled in this course. Please join this live sessions because these live sessions are basically organized for you for your benefits only. We'll be discussing throughout these 2 hours is whatever has been taught in week one. I'll give you some more applications about the topics that has been taught, and on top of that, towards the end of the lecture, I'll be solving some MCQs and multiple answer correct questions, which will help you to do your assignments. So. Uh, yeah, please do join this session every Tuesday, 6 to 8. And now let's get started. So I'll just share my screen here. Okay. Yeah, and I hope you can see my screen. The name of this course is Computational Systems Biology. This is taught by Professor Karthik Raman of IIT Madras, and I'm the PMRFTA for this course. My name is Shoma, and I'm from IISc Bangalore. I'm a first year PhD student, and let's get started. Before I move into specific topics, specific concepts that has been taught into week one, I would just like to give everyone of you a summary of what basically systems biology is, and like how it helps us uh, say, transitions or different kind of biological mechanisms going on inside the cell in a theoretical manner. Obviously there is a experimental approach to it as well, where you use omics data, and you it's kind of feedback model that goes on. So you have omics data, you use that to create models, create networks, create pathways, and then you model them, and then you have some sort of hypothesis coming out of the model, which you then apply into the experimental experimental setups that you are using to prove your hypothesis, so that you can have better understanding of what is going on before even you do any experimental work. So basically, this is a paper that has been this paper got published in 2002, and uh, this is I find this paper a very good example of a very good document of how and why we use systems biology in biological systems. So I suggest everyone go and read this paper. The name of the paper is very simple. It's kind of biologist fix the radio. It's not a technically it's not filled with many technical details. It gives you a very good overview about why we do need systems biology, or why we do need systems level thinking, at least not to understand such complex systems. So let's get into it. This paper is written by a person called Leslie, and I have made a summary of this paper through 5 points. Obviously, this does not cover the entire paper, but if you want more information, I suggest you go read it. But these are the five major things that he talks about in the paper. So first comes the need for new languages. So biological methods focusing on individual components and their interactions are insufficient for understanding complex phenotypes like cancer. We need new languages that can describe that emergent properties arising from these interactions. So you may notice that I've used the term complex phenotypes like cancer. So uh, cancer or any other disease that is multifactorial in nature, so that you cannot pinpoint a single reason to which the disease is occurring inside the system, on the body or the cell, uh, there are multiple factors involved in complex diseases like cancer or say neurodegeneration, or there are many examples like this. So we need to since these are complex multifactorial diseases, there are no single molecules or no single gene that is solely responsible for causing this phenotype. There are a multitude of reasons that is responsible. And like say for example, you have a you have the P53 gene in your cell, that is the tumor suppressor gene. So it can get mutated. If it gets mutated, like the signaling pathway that it follows, that also gets hampered. So if it goes and binds to some other factors like say a P38, it will not be able to bind if it is mutated. P53 is mutated, so it'll go cause disruption. It will go basically not disrupt your cell cycle, but you lose regulation over your cell cycle processes, which may lead up to cancer. I'm talking about a single path, but it's not as simple as, of course, many of you know. Uh, you need multiple such reasons to get a phenotype like cancer, because our body or the biological system, it's very resilient. So a small perturbation is not enough to get a complex phenotype. So you need to perturb the system enough to get that phenotype, and that is not possible through a single molecule or a single gene or a single pathway. So individual components and their interactions, although are necessary. Um, just a second. I got disconnected. Yeah. Sorry. So yeah, we need new languages to better understand these interactions, this the how the individual components are affecting each other. So that new language for us is mathematics or computation or whatever you theoretical sciences, whatever you want to call it. But yeah, we need new new languages to understand these complex phenomena. Next is the importance of connections. So complex systems are defined by the intricate connections and feedback loops, not just individual parts. Analyzing these connections is crucial for understanding their behavior, which is very true, because if you look at how the cell works, it, it has a very well regulated system. So there are signaling pathways, there are different transcription factors that are controlling and regulating every point of of this, basically of any activity in the cell. So any activity in the cell will be highly regulated, and they will have intricate connections and feedback loops. So uh, uh, so that you can have a better regulation over the process. So not only are the individual parts important, how these individual parts are connected and how they feed back into the system, and how they regulate each other. That is a very important part of how biological systems work. So understanding these connections is very important, because without understanding these connections, if you just focus on a single molecule or a single gene or a single signaling element or a protein, you may miss out on these on the uh, overall behavior of the cell, how it is coming to that behavior. So you need to understand how these uh, elements are connected and how they regulate each other. So that is the second point. The third point is the limits of reductionism. So breaking down a complex system into parts can be helpful, but it can also, like, you may also miss out on the bigger picture. We need to recognize the limitations of reductionist approaches when dealing with intricate systems. So that is what I was talking about. So basically, a single component or a single individual um, element in the cell or the system, uh, like say for example, a particular gene, is mutated for a particular disease. So, say for example, you want to target a drug so that you can have basically a cure for the disease. So you'll think of targeting it into the gene, but it obviously it may work. But in maximum cases, is what will happen is that gene is regulated at different levels in the system. So it is not say for example, the transcription factor is not allowing the gene to transcribe properly. So what you do is you take a drug and try to like induce the transcription of the gene. But the point is, it may work, but in most cases, it may not work because it is not the only component that is contributing to the phenotype. You need to look at the bigger picture of how that component is responsible, and also how other components are also responsible for the phenotype. So reductionism has its limitations, but and if you do it too much, you may miss out on the bigger picture, which will not help you do your work at all. And coming to the next point is the value of systems level thinking. So understanding complex systems requires a holistic approach that considers all parts and their interactions, not just individual components. This systems thinking approach is essential for comprehending how the whole phenotype emerges from this part. So yeah, this is it kind of summarizes of how what we were talking about in the last few minutes, that uh, you need to have a systems level thinking, not just focus on individual components. Uh, so yeah, and the last, and this is a very important point, is that complex systems are often influenced by chance events, but they respond to these events depending on their underlying structure and preparedness. Understanding this interplay between chance and preparedness is crucial for predicting systems level behavior. So, like, a complex system you uh, will have uh, chance events. Basically, uh, you can have chance events that can contribute to mutations. You can have chance events that can contribute to, say, overexpressing of a gene, or you can have chance event that can, say, like, knock down a gene also. So the inside complex systems, these are very much possible. Mostly the systems are under very tight regulation from various different molecules, various different pathways and all. But chance events are not something that is uncommon in biological systems. So understanding this interplay between what is a chance event and what is preparedness of the biological system is crucial for predicting the system's behavior. So if we move on to the next part, so this is where we discuss that why we use mathematical models. So in the last slide, we discussed why we need a systems level thinking, and the systems level thinking requires basically a language to basically communicate. So that language is mathematics, and so we use mathematical models to predict that systems level behavior. So why we use mathematical models? First is models articulate our conceptualizations of how the world operates. So you can have very complex phenomena that are occurring, but that can be very easily easily written down in terms of mathematics and differential equations and models, or uh, which can like theoretically simulate how that system is working. So it serves as a conduit, translating the beliefs, the understanding that we have into precise language of mathematics. So which is very important. And what are the advantages of mathematical modeling? So first thing it is the precision of mathematics. So it is a highly precise language, and facilitates the formulation of ideas and the identification of underlying assumptions. So uh, obviously like it is a highly precise language. You can like write down, as I said, you can write down complex interactions, complex phenotypes emerging from a system in terms of a set of differential equations or a set of other equations, not only differential equations. Say, for example, you are working with a metabolic network. So how a particular metabolite is getting formed, you can model that. That may be a very in itself the formation of the metabolite inside the system. That may be a very very complex what to say a very complex uh, phenomena, but in terms of mathematics, you can write it down in maybe like 6 7 equations or 10 equations or whatever. Basically, what I'm pointing to is the complexities can be broken down, or the complexities of the systems can be broken down in terms of simple mathematical equations. Next thing is leveraging centuries of mathematical insights. So basically, mathematics has been here for ages, from the time we know science to exist. So mathematics is a very old language, and it has been uh, highly, like, what to say, refined over the years, over hundreds of years by several mathematicians. And you have a lot of what to say, a lot of knowledge of mathematics that you have currently. You can use that to enhance and refine your models. So yeah, not only is is it a precise language, it is also very common and very, what to say, very old and accurate language. To say. Yeah. And the next thing is computational powers. So what happens is, if we take the example of any network, simple, even to a simple like 2 or 3 node network, we if we try if we consider that three node network, and we try to find out how these 3 nodes are like are helping, basically are contributing to a particular phenotype. uh, uh, Basically, if you want to model that, you'll be doing that in terms of differential equations. So if like the guys who have a little bit of mathematics background know how difficult it is to solve differential equations, right? So it is very rare that you can get an analytical solution. So like it's not it's in 99.9% of cases, it's not possible to solve those equations by hand. So what you need is large computation powers. So you need you have programs like MATLAB, where you like feed the equations into the program, and it will solve it for you with numerical methods, which is not actually, like, I would say, obviously it is possible to do it by hand, but it is very tedious and it is not at all practical. So for complex systems, you'll have huge number of equations. So solving those equations by hand is not not feasible, basically. So you can, but you have other tools to help you do that. You have programming languages which will solve it for you numerically, and you can leverage these computational capabilities to simulate and analyze this complex phenotypes. Just a second. Okay. So next comes the steps of using mathematical model. So we learned about what is systems biology, what is systems level thinking. Next, we learned about why we use mathematics as a language or mathematical modeling as a technique to understand the systems level behavior of complex phenotypes. Now, let's come to the steps of using these mathematical models. So, first and foremost, and the most important part of it, is identifying the need, defining your research statement. So, that is the most important part of mathematical modeling. You need to have a clear and sorry, you need to have a clear definition of the purpose of the model and what it aims to address. So that needs to be clear first and foremost. So you need to have a clear research statement that you want to do this with the model. You want to predict some, you want to predict, say, for example, for like the example that is talked about before, you say, for example, you want to predict how a metabolite is getting formed. So depending on your research question, uh, you will basically build the model. The model doesn't like it doesn't work the other way around. So it doesn't work like this, that you first build the model, then you identify what to answer. It's actually the other way around. So you identify what to answer, and then you build the model accordingly. So yeah. Next is the parameter or variable listing. So the model will have a lot of parameters. If it is a big model, it will have a lot of parameters and a lot of variables which are required for the model to run, and different values of these parameters and variables, and different what to say different utilization of these parameters will give you different results. So it is very important also to identify which parameters are key, and which variables are key, and which are required for the model and which are not required. So this is kind of you can think of it as a pre-processing step before you actually build the model, that you identify which are the important parameters of this model, and which are the important variables. Next comes the data recognition. So I will talk about this a little bit further down the lecture, in this lecture only, but I'll give you an overview. So, okay, just a second. Sorry, guys, I'm having a little bit of internet connection issue. I think, yeah, now it is working. Okay, so next comes the point of data recognition. So, as I said, I'm going to talk about this in a little bit more detail during the later part of this lecture. But in general, uh, like recognizing the available relevant data is very important. So majorly the kind of data that we use for this kind of modeling, this kind of mathematical modeling, or any systems biology methods is basically omics. So we have different kinds of omics data. We have genomics, we have transcriptomics, we have proteomics. Now we have multi-omics. So you have genomics, proteomics together, genomics, transcriptomics together, or there are variety of combinations. And uh, basically these help you to delve into a vast world of data. So uh, identifying your what data suits your needs, your research question, and your model, that is very important. And identifying the circumstances and assumptions that can be applied can be can be inferred from the data. So the data recognition and the data identification is very important. So next comes the governing physical principles. So basically, if you talk about, for example, a network, so say for example, a metabolic network, or I've been talking about metabolic networks. I've been giving my examples of metabolic networks too much. So I'll talk about, say, gene regulatory networks you have, or transcription regulatory networks. So these kind of networks will have in the nodes, as the like in the nodes you'll have the different molecules or the genes. For example, in gene regulatory network, the nodes in the network will have different genes, and the edges will denote how they are interacting. So if it is getting upregulated, if it is getting downregulated. So these interactions between different elements of the system is governed by varying physical principles. So it may be a repression, it may be a overexpression, it may be a, say, a knockdown. It can be anything. So identifying these governing principles and implementing this in your model is what makes your model good. So the more information of you have about how these individual elements interact with each other. This was the point that I was talking about a couple of slides back. That identification of like interactions between the individual elements in the model, it is very important. So like, that comes from like, and how would you get that? How would it know what physical principles are governing? So there are various ways to find out. Like first and foremost is you go and do literature research. So obviously this kind of data, this kind of information is only possible to have from experimental techniques or experimental data. So you need to know beforehand that this component is, say, inhibiting this component or overexpressing this component. So that that connection and the type of connection is very important in your model and how your model behaves. So governing physical principles, you need to know beforehand, or you need to find out before you build your model. And the more information you have about how this how these interactions are occurring, and how the physical principles are governing the particular say reaction or a particular state or a particular production of a metabolite or whatever it is very important for your model to perform well. The more information you have about how these physical interactions are occurring inside the cell, the more realistic your model is. Basically the more realistic the model uh, uh, basically the more realistic is the results of the model. So yeah, next comes equations and calculations. So yeah, as I talked about, for example, uh, we use differential equations to solve the various models, and we do calculations and solve these differential equations and find out a steady state or a transition or etc. So as I said, like, you cannot do these calculations by hand. Maximum times you'll need a computer, you'll need a program to run the calculations for you, which is not a bottleneck now because you have enough computational power to run this kind of simulations. So yeah, that is the fifth point. And the last, this is also a very important point, is to check for the consistency of the model and the unity of the model. So your model should not break down. So as I was talking about in the beginning, that complex systems are very resilient in nature. See, you cannot just shift a state in a complex system by just doing a simple perturbation, because it is resilient. It will come back to its steady state. Uh, you need enough. Uh, you need enough perturbations to actually move the system from its steady state or its previous state. So your model should actually mimic that behavior. Your model should also be very consistent. Your model should not give you random results, and your model should have unity. So yeah. Okay, moving on. Next comes biological model. So till now we have studied what is systems biology, why we use mathematical modeling, the steps of using mathematical modeling, and now we come to biological models. So, understanding complex biological systems required more than just characterizing individual molecules, and it involves comprehending molecular interactions and pathways. So, as I was talking about, so you have, you take any pathway in a system that is any signaling pathway in a system, in a biological system. It will have various components. It will have a lot of molecules involved, a lot of genes involved, transcription factors involved. It's basically a big system on its own. It is individual signaling pathways. So it's very important to understand their interactions and how they're getting regulated, how how they're how they're kind of forming feedback loops, be it positive, be it negative, how they're like helping the system to stay in that steady state or stay in that particular state. So not only single individual elements, which may be individual molecules, individual genes. It is more important to know the bigger picture. So I like I told it wrong. Uh, Not more importantly, but it's as equally important to understand uh, these to interactions and how these interactions are leading to that particular thing. So the next point is mathematical models play a crucial role in investigating how regulatory processes are connected and how disruptions can contribute to the disease. So yeah, this is basically what I was talking about. You'll have different kind of regulations in place for every element inside the system. So it is, and mathematical models can help you understand and investigate how these regulatory processes are interconnected and what kind of disruptions, not only how and what kind of disruptions can contribute to diseases. So it's kind of you can predict from previous data and from your model that yeah, say maybe in a particular pathway, this element, if it gets mutated, it will not bind to this molecule, which in turn will not do this function, which in turn will say, for example, in like remove your cell cycle checkpoints, and you can go on in the cell cycle forever and forever, like which can form a tumor or a cancer. So like it is important to know how these regulatory processes are interconnected, and these mathematical models can help you find this information. Next comes computational models. They are also uh, they are also very what to say now competition with the increase of computational power, so computational and statistical models like the machine learning models that we have right now, regression models, or say like we now have deep learning models as well. So these are sophisticated models which can assist systematically analyzing system perturbations, guiding experimental design, and evaluating molecules as therapeutic targets. So I have actually discussed about all the three uh, functions that is talked about in this point. So systematically analyzing system perturbations. So what kind of perturbations can form diseases? What kind of perturbations will not form diseases, or not diseases particularly, but any kind of phenotype, not only disease. So it is very important to understand uh, these perturbations and how they can affect the system. Next is guiding experimental design, as I talked about. It's kind of a feedback loop. You have your data, you kind of understand the physical governing principles that is going on between individual elements in the system from the data. You then put it in a model, you model it and you simulate it, and you get some kind of hypothesis and results, which can again help you build a better experimental design, which will again give you a better interactions between these elements, and then you get a result, and then it continues as a loop. So uh, basically data is important, and so is the model, and they basically feed off each other. And the last part is evaluating molecules as therapeutic targets, which I actually talked about a few minutes earlier. Yeah, you have what to say you can you can you can find therapeutic targets with the help of mathematical modeling. As I discussed, if you know what kind of perturbations are causing this and what kind of perturbations will cause a particular element to not interact or to not do a particular function, you can identify what exactly is going on in the system, and uh, you can target your drug specifically to the particular molecules. Next comes that various mathematical models address different biological processes from metabolism to signaling pathways. So there are if you go and search biological networks, you'll find a variety of biological networks. It can be a disease network, it can be a metabolic network, it can be a gene regulatory network, it can be a transcription regulatory network. So there are different kind of biological processes, but you can use mathematical models to address these all different kind of biological process from metabolism to signaling pathways. So anything you can think of, you can just you can just model it according to their interactions. You can write a few differential equations. You can write a few equations in general, and then you can solve them and try to find out what kind of stable states they are in or what kind of transitions they are in, etc. Next comes is that mathematical modeling is essential for biologists to understand and analyze physiological processes and for pharmaceutical industry to support drug discovery and development. This is basically what I talked about in the last couple of minutes. That yeah, you can identify these drug targets from with the help of modeling, and uh, you can understand these physiological processes and how your particular drug target is affecting these physiological processes and how you want it to behave. So you kind of can predict these behaviors from models, from essentially mathematical models, which can help you uh, support drug discovery and development. And the last point is the the data that we talked about, omics. So omics is a vast, like what to say, it's a vast collection of data, you may say, but it's a technology. It's not a data set or anything. It's a technology, and it has different kinds of data under it. So if you do DNA sequencing, that is genomics. If you kind of do mRNA sequencing, that is transcriptomics. If you you can do DNA sequencing using basically you from DNA, then comes RNA, then comes protein. So that is the central dogma of biology. But you can from from DNA to RNA to protein, you can at individual levels you can uh, you can investigate how these how various processes work. So you can have you can have DNA sequencing, you can have RNA sequencing, you can have proteomics techniques such as MS spectrometry and other techniques as well. So at each level, you can have that information of how different elements are interacting with each other and how they're regulating with each other from the data. So recent advancements in omics technologies have vastly contributed to allow us to monitor cellular processes at the molecular level. I will come to omics a little bit later, but yeah. So, what exactly are the kind of like how does biological models work basically? So this picture on the right gives you a fairly decent overview. So you have a biological phenomena, and you 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 understand that through experimental data. So you have a biological phenomena. Say you do RNA sequencing, and then you understand कि, in this particular case, in this particular phenotype, a particular gene is not expressing as much. It is not getting expressed as much in this particular phenotype. So, but that you can include in your mathematical model, and the results of the mathematical model will give you a hypothesis about how the phenotype is getting generated, which can help you design new dry and wet lab experiments. Uh, you can design new computer simulations, or you can design new laboratory experiments and get new data in support of your logic, and then feed it into the model again, which will give you an even better hypothesis if you're obviously if your the fed data is correct and accurate. It will give you a better the model will give you a better hypothesis, uh, which in turn will give you a better uh, better idea about what kind of experiments to conduct, be it dry or wet lab. So it kind of moves as in a feedback loop. So you have biological phenomena, you have experimental data, then you have mathematical models. You get a hypothesis from that model. Now, with the help of that model, you design these experiments, which generate new data, which you and inference from the data, which you can feed back to the model. Next comes sorry. So I'll go through this in a minute. So Biological systems with numerous molecular components exhibit emergent properties. So, uh, like the biological systems are complex, and with several numerous molecular components, it can there are a plethora of possibilities of plethora of properties that it can exhibit. So, uh, but you can see emergent properties from biological systems. Nonlinear reaction kinetics and complexities like cell structure contribute to this unpredictable behavior. And also, mathematical models capture these complexities, allowing in silico simulations. So, these complexities that we talked about, these kind of interactions, this kind of uh, like binding or regulations that we talked about, that can be elegantly captured in mathematical models. These complexities can be elegantly captured in the models, and that can allow in silico simulations. And computer basically dry labs, dry experiments are basically cost effective because you're running them in a computer, which is cost effective and even less time consuming compared to wet lab experiments. So. Uh, uh, your computer simulations will be obviously less time consuming than your wet lab experiments, but your assumptions and your understanding of the system needs to be good to get good results. You cannot do random simulations and expect good hypothesis or good results. You need to be very careful about the kind of interactions that you are introducing into the model, the kind of parameters that you are using in the model. That has to be correctly identified so that you can get a good result. Models predict systems behavior and systematically investigate these perturbations induced by various factors. And that factors may be anything like I talked about. So, simulations are less constrained by experiments and allow quick exploration before laboratory assessment. So, before you want to prove or disprove a hypothesis using wet lab techniques again, because at the end of the day, like you need to prove that this is actually what is happening inside the system. So wet lab techniques are obviously very important for that. So if, before you even design a wet lab experiment, these simulations, this mathematical models and the results can give you a good hypothesis about what is happening inside the system, and according to this hypothesis, you can design these experiments better. And the combination of this dry and wet lab approaches in systems biology aids in this hypothesis development. Okay. And in metabolic engineering, models suggest genetic modifications for enhanced productivity. And models help identify simpler subsystems sufficient for understanding overall systems behavior. So for example, you have a very big metabolic network, but you are only interested in finding out how a particular metabolite is getting formed inside the network. So maybe you do not need all the interactions, say like a 5 million interactions in the network to understand how that particular metabolite is forming. Maybe that can be explained by a set of like 10 or 15 or 20 or say example, for example, 100 interactions. That is obviously much lesser than like millions of interactions. So you can identify smaller subsystems inside the system to cater to a particular question of a particular molecule or a particular gene or a particular signaling pathway. Next comes steady states. So before I move into steady state, can like I would like all of you to answer which of these three states, S1, U1, and S2, are the most stable. I'll give you a minute. Okay. So if you look at this Uh, Imagine these three are balls, and this is a mountain terrain, and on this terrain there are three balls. Which one do you think is the most stable? So if you see y-axis as the energy component, then S1, S2, and U1 are on different energy levels. So U1 has the highest energy level, followed by S1, followed by S2. But there is a fundamental difference between S1, S2, and U1. See, if you in either of S1 and S2, if you try to perturb this system, say for example, you try to move the system towards the right or towards the left. So you try to move S1 towards the right or towards the left, it will come back to its original position because that's a steady state. Similar is the case with S2 because this is the lowest energy point in the vicinity. So even in case of S2 as well, if you try to move the ball up in the right or in the left, it will come back to its original position. But in the other case, U1, it is stable, but sorry, it is at a steady state because it is not moving, but it is not stable because if you perturb it a little, be it to the right or to the left, it will not come back to its original position. It will slide over to either S1 or S2 depending on which direction you push. So these two states, where you perturb the system and the systems come back to its original position, are called stable states. And the one that does not come back to its original state when perturbed is called the unstable state. So you can have stable steady states, you can have unstable steady states. Now, steady states is something that basically is defined as where there is no change in the system. Basically, if the phenomenon is given by n, basically d and dt equal to zero. The n is not changing with time. In simpler terms, the systems isn't changing over time. So all three of them are steady states if you look carefully because these are like if you do not perturb them they won't move, so they are steady states. There is no change in the system that is occurring over time. So these are stable states, steady states, but these are not, all of them are not stable steady states. So S1 and S2 are stable steady states because if you perturb them it will come back to its original state. But U1 is an unstable steady state because if you perturb it, it will not come back to its original state. So stability can be stable. Basically, steady states can be stable or unstable. As I defined, if a small disturbance away from the steady state causes the system to return to the steady state over time, then the system is stable. The steady state is stable. And if a small disturbance causes the system to move further away from the steady state, then the steady state is unstable. Okay. Uh, there is another component to this, which is called attractiveness. So, sorry. Ah, so, if any small perturbation leads the system back to its steady state, then the steady state is attractive. It is like a magnet pulling the system back in. So, S1 and S2 are attractive steady states. If you move it to, if you try to move it, it will pull it back. And another point is repulsive steady state. So if small perturbations cause the system to move further away from the steady state, then the steady state is repulsive. So basically, these states are attractive, and this state is repulsive because if you push it, it will move away, it will not come back. Another few terms that I wanted to discuss before moving on to different examples and applications of systems biology. Um, uh, the couple of terms I want to introduce are robustness and resilience. So, what is robustness? Robustness is the ability of a system to remain in or reach the same stable state. So, it's basically this is ability of the system not to perturb itself. It maintains original state despite diverse internal and external environments, and emphasizes a biological system's resistance to perturbation. So, as I said, uh, believe the biological systems are the most complex systems in one of the most complex systems in nature, and robustness of biological systems are very important in how we live. So, say for example, we live in a world where everywhere around us there are carcinogens. There is carcinogen in sunlight, the UV light. There is carcinogen in airs, through pollutants. There is carcinogen in your food, in vegetable colors, and everything. There are there are basically, if you want to call them perturbants, elements that can perturb your system all around you. But we do not all have cancer, do we? But technically, we should have, because all the carcinogens are around us, all the elements that are looking to perturb our system is around us. But that is the beauty of biological systems, that it is very, very robust in nature. Small perturbations are not capable of developing a phenotype or moving this uh, the stable state of the biological system. Next comes resilience. So resilience is defined as the ability of the system to return to a previous state or establish a new state after significant perturbations. So basically, if you try to move a system from one state to another, and if it if it if it can establish a new state after significant perturbations, and that state is stable and doesn't move away from that, and basically it stays in that state, it's the, basically the higher of resilience you'll have, it the higher is your ability to return to a previous state or establish a new state after significant perturbation. So it basically gives an idea about how the system is adaptable and how the system recovers in face of perturbations. So these are robustness and resilience definitions. I have given a little bit examples of what you can call robust and what can you call resilient. So first is these are examples of plants. So basically, if a plant is both robust and resilient, it would grow normally across all light conditions. Say the plant is resilient but not robust, it will become dormant in the dark, but it will rapidly grow in the desired light conditions. If it is robust but not resilient, it grow under most light conditions, but will struggle while switching between them. And if it is neither robust or resilient, it will grow under only one specific light condition and will die when that condition is gone. So resilience focuses on your system's adaptability and recovery capabilities, and your robustness focuses on your system's resistance to perturbations. Moving on. So this is something that has been discussed in the course lectures as well. So while implementing models, you'll have different kind of basically what is overfitting and underfitting when you're like fitting a model to a set of data. So this is not only true for mathematical models. This is true. Basically, this the whole uh, plot that I have, the picture that I have put here, the equation also that I have put here, is basically taken from the concepts of statistical models, but the concept is same in mathematical models as well. So if you look at this equation, this tells you this is basically the expectation of y star minus f x star given d whole square. So basically, your y star is your original value, and your fx star given d is your predicted value, and you are trying to find the expected loss of your model, basically in terms of prediction and the ground truth. So that can be written down as, this is a complex mathematical proof, I won't be going down here, but overall it can be written as noise plus variance plus bias squared. So the loss is dependent on both your bias and variance, and your bias and variance need to be, there needs to be a trade-off to have that ideal uh, optimal model complexity. So basically, if your model is trying, say you have a data set and you want to fit a model. So, and if your model fits each and every point of the data set, that model is overfitted on the data. So basically, you have your data set, you fit your model through each and every point of the data set. That model will be a very good representation of that current data set. But if you want to use that model to predict some other data, that will not be possible, because the variance is very high in the previous data set, and you have fitted the model up with that variance. So you have real world data, which you get from real world, that has quite some variance. If you fit a model along with that variance also, your model is not a uh, good explanation of the of the whole basically system, because it has more fitted to the variance than actually how the data is actually how the system is working. So it is more fitted on the variance than on the bias. So, and even if your bias is too high, it'll like that is also very bad. It'll underfit the model. So the data, basically, if your bias is very high, your model will not fit to maximum of the points, and it will not be able to explain maximum of the phenomenon. So there needs to be a trade-off between bias and variance. Your bias also cannot be too high, your variance also cannot be too high. So ideally, this is how the bias variance plot looks like. So this is the red line is your bias, the the blue line is your variance, and this is your total error. So the total error will be lowest at this point, where you kind of you have a trade-off between bias and variance. Not Neither is the bias too high, nor is the variance too high. So you need to find that optimal spot so that you do not overfit or underfit your data. So next, I'll be talking about a few examples, and basically a few applications of modeling in biological systems. So this is one of the models that has been here for a long time. So this has been here for, I think, till from the 1950s, and this model has stood the test of time. This is the gate model proposed by Hodgkin and Huxley. Basically, if you guys know a little bit neuroscience, so what happens in is your basically you have your neuron, you have your dendrites and your axon. So axon is the place where your action potential starts. Basically, your signal starts and it flows through whole of the axon through the synapse, and it moves on to the next neuron. So these these signals that are passing through the axon, these are generated within the axon called as action potentials. So these action potentials are generated by a complex mechanism of both utilizing both concentration gradients between different ions and also charge gradients between different ions. And there are voltage gated channels on the walls of these axons, which open and close according to the voltage, which allow um, uh, molecules to come in or go out, and uh, perturb the total, basically the voltage across the membrane, which can form, and if there is a voltage difference at one point of the membrane compared to the other, a current will flow. So basically, these channels are very important to create create this voltage and help the action potential flow, or basically the signal flow through the action. So these ionic channels in the membrane undergo conformation changes in response to radiation and electric field, and this conformation changes causes the channel to move from open to closed states and vice versa. Now, if you look at this, basically this graph or diagram, whatever you want to call it, so y is your permissive state, and 1 minus y will be your non-permissive state. So these are called, like, they did not know what actually was happening inside, what actually ion channels are, or how our ion channels actually are working, like the, basically uh, the method by which they are opening and closing, the reason behind it. So, but what they what they like described are gating particles. So basically, if your gating particle is at this non-permissive end, it will not open. If it is at this permissive end, it will open. So they imagined these two states between to transition between each other. So A goes to B and B comes back to A. This is basically a chemical equilibrium, if you recall. So you have this delta G naught, which is the activation energy of the particle. So if this particle can move from here to here, that it needs to cross this energy barrier. But what happens is when the voltage is applied, this energy barrier, you can see with this dotted line, this energy barrier is a little bit lowered, which happens if you like remember your class 11 and 12 chemical equilibrium. uh, this is how actually enzyme catalysis works. So enzyme catalyst, what it does is enzyme brings down the activation energy of the whole reaction so that it can easily move from one state to another. So this is also a similar concept here, but here the electric field or the voltage difference is causing this change in activation energy, which is given by this del Z Fe, and you can and the gating particle can move from this state to this state. So non-permissive, it can move from a non-permissive to a permissive state. So and now you can like this alpha V and beta V. These are basically the reaction rates of open to close and close to open, and you can write this in form of these equations. This is basically ae to the power minus ea by rt form, and instead of minus ea, which is activation energy, you write del g naught. This is the del g naught, and using uh, basically the electric field, it has been brought down by del z fe, so minus del z fe, and similarly with beta v. So you can uh, find these reaction rates, and you can you can like model this whole opening and closing, moving from one state to another. There's a simple first order kinetic reaction. So dy dt will be alpha into 1 minus y minus beta y, and at steady state is no movement in the system, so it will be zero, and therefore your y infinity, which is basically it is denoting at infinite time. You'll have this, basically it is imagined that you'll have the steady state at infinite time, so that y infinity is given by alpha by alpha minus b, and you can have these kind of plots. This is a very old model, but this is a very crucial model in understanding how basically neurons work and how action potentials are generated and traveled and propagated. And this has stood the test of time. Not many models have. This is one of the very few in biology that has stood this test of time. I strongly suggest you go and have a look at this model. You can Google it, or if you want a detailed mathematical representation, or now how exactly is this model working. These figures have been taken from a book called Johnston and Wu. This is a neuroscience book. You can go and take a look. You'll find a lot of interesting stuff. Okay, next example is that of NF kappa B. So NF kappa B is a protein that uh, plays many roles inside the cell. It is involved in inflammation, it is involved in immunity, it is involved in cell growth, survival, anything, you name it. And then if kappa B is there. But what happens is it is normally found in the cytoplasm, but it moves into the nucleus to basically turn on genes. So basically, it acts as a transcription factor. So it travels from the cytoplasm to inside the nucleus into the chromosomes and binds on particular genes and acts as transcription factors, facilitating their transcription, the target gene transcriptions. Now, to prevent NF kappa B from moving into the nucleus inappropriately, it is often bound to inhibitory proteins like I kappa B. So basically, what happens is when you have to move across across the nuclear membrane or the nuclear envelope, you need to be of a particular size, or else you will not be able to get in within the pores of the nuclear envelope or the nuclear membrane. So it, I kappa B binds to NF kappa B, which does not allow that the complex to get inside uh, the nucleus. So, but when the signal tells the cell to activate NF kappa B, the I kappa Bs are destroyed, and then the NF kappa B can easily move into the nucleus. Now, if you go inside, one of the genes that NF kappa B turns on is actually a gene for I kappa B. So I kappa B is turning off NF kappa B outside, and now NF kappa B is coming inside and turning on I kappa B. So it's kind of a negative feedback loop. The cycle of activation and inactivation helps to keep the NF kappa B levels in check. It also helps like regulate how much activations of particular genes that are that are facilitated by NF kappa B are, are occurring, and how the expression is changing, that can be completely regulated using these negative feedback loops. And scientists have used mathematical models to study these to study the way NF kappa B is regulated. I'm sorry you cannot see this, but these models have helped to identify other molecules also that have a role in uh, regulating NF kappa. So if you look at if you look at this if this pathway, this is actually basically you see TNF coming in and basically starting the signaling cascade, and this is basically a little bit complex cascade, and you can write down this in differential equation forms. Now you do that, you find basically you find you have results which you implement in your experimental analysis of NF kappa B, I kappa B activity over time. And you see that it becomes smaller and smaller over time, and in agreement with the predictions of the mathematical model. So this temporal behavior of the pathway can also be expressed, and basically detailed analysis of the experiment data and the resulting graphs and what is happening is basically you can feed that information back into the model, which can lead to further refinement of the you can look at these sources I've mentioned here. This actually explain a little bit more on how NF-kappa B signaling is actually regulated and how you can model it. I'll suggest, if you are interested, please go and have a look. Okay, so this is the last slide of my lecture. After this, we'll move into assignments, basically question solving. So this is the overview that I was talking about from the beginning. So you have on the left side, you have basically genomics. Basically from DNA, what information you get, then you integrate that information with transcriptomics at the mRNA level, how your genes are getting expressed, and then further down at the proteomics level, how your proteins are getting expressed, how they're getting formed, how they're binding to each other, etc., etc. And you use all this information to form networks that can be transcriptional, that can be metabolites, that can be interactomes, metabolomes, whatever. You can form these networks, and you can use mathematical modeling to form design principles to not form, to predict design principles of further experiments that you want, that you like basically that will help you prove your hypothesis. You can find emergent properties, circuit logics, modular organization. Along with the phenotype data, you can get a total idea in a theoretical manner about how the system is working and how it is interacting with each of its components. So on the extreme left, you have genomics. After that, you come to transcriptomics. Then you have proteomics. Then you integrate all these three into networks, which you further model and get ideas about design principles, emergent properties, etc., etc., etc. This is where the systems biology part lie. and this is where the omics part lies. So basically, we omics and systems biology go hand in hand, and uh, currently these are very important to understand how the systems are working from be it from a wet lab perspective or from a theoretical perspective. It is very important to kind of continue this feedback loop of getting information, refining a model, get doing better experiments, and getting information into the model and refining the kind of the feedback loop that we are familiar with. So yeah, any questions? Okay, so we'll move on. I'll what will happen is I'll keep these questions on the screen for 2 to 3 minutes, and after that I'll show you the answer and I'll explain uh, the answer. So I suggest you look at this question. You have 3 minutes to answer each of the question, and after that I'll give you the answers. Okay. So the question was, which was of the following is true for a mathematical model? So first option, all models are wrong practically. That is very true. So basically, this it's a theoretical representation of what is happening in a system, and there are a lot of assumptions that comes with it. And yeah, it is what makes the models wrong. Basically, there are no correct models. All models basically are wrong. So next point is that mathematical models are only true for the assumptions that we make, which is very true because you build your model, you build your connections, you build your equations, and you solve them according to the assumptions that you make. So the the results you get from a mathematical model, the kind of hypothesis you get on the mathematical model, is conditional upon the hypothesis or the assumptions that you make before building the model. So if and only if that assumption is true, then only that model will give you a good uh, good result, or else it won't. So yeah, it is only true for assumptions that we make. Never overfits if you consider large data. That is actually not true. It is very easy to overfit. Actually, overfitting or underfitting a data set of a model on a data set, it is not dependent on the size of the data. So a model can overfit a large data, model can overfit a small data as well. So it does not depend on the size of the data. Next is complex models always perform better than a simple model, which is absolutely not true. So if your phenomena, if your ideas, or if your model basically if your interactions, or if your uh, uh, if the states that you're defining inside the model are simple, basically, if you can do that using, if you can model a particular phenomena using a simple model than a complex model, it is actually better to do it with a simple model, because a complex model does not give you that guarantee that will perform better. So it will never give you that guarantee. In some cases, obviously, it will perform better because it will have more information, but it will never give you that guarantee that it can always perform better than simple models. So uh, initially, it's better to use simple models, which gets you a better understanding of this whole modeling process than to use complex models. So, yeah. And a last point is simple models may be preferable under certain circumstances. That is actually very true. So if you have a very large network and if you have a lot of components inside the network, bringing unnecessary complexity inside the network, from like hypothesizing about an unnecessary complexity inside the network will not be beneficial in terms of getting results from the model. So simple models is preferable under certain circumstances, because complex models does not give you any kind of guarantee that will perform better. Before I move into this, I just want to quickly check if the recording is going all right or not. Yeah, it looks all right, I think. Okay. So let's move on to this. The next question. This is also multiple answer correct. Just to give you a heads up, if you see these square boxes as options, it is multiple answer correct. And if you see round boxes, that is basically single answer. So yeah, I'll give you 2 minutes. Okay. So the question is that you are asked to build a model to predict max and minimum temperature for the city of New Delhi. Data collected from the year 2000 to 2015 is given to you, which measures different parameters such as humidity, solar radiation, pollution, air velocity, pressure, etc. And you are using a polynomial regression model by reducing the RMS error. And the model you built is first is mathematical, which is true. It is a mathematical model because you're trying to predict the maximum and minimum temperature of of the city. So it is indeed a mathematical model. It is deterministic as well, and it is empirical as well. What it is not is stochastic and discontinuous. So it will not be stochastic because you cannot predict statistical outcomes of this model, statistical outcomes of the data from this model. and you can, and it is not discontinuous in nature because you have continuous data from like 2000 to 2015, so there is no discontinuity. It is empirical, it is deterministic because you're finding something, and it is obviously mathematical. So these three answers are correct. Next question. I'll give you 2 minutes. Okay. So the question is, biological systems are mostly, this is also multiple answer, correct? So sensitive, is it? No, because biological systems, as I said, are very robust in nature, very resilient in nature. It is not sensitive to small perturbations. It can maintain its own state, and it is not sensitive at all. Stable, obviously it is. Biological systems are stable, that's why we are living. And uh, yeah. So, and next, consistent. It is not consistent because there is a lot of heterogeneity inside the systems, so it is not consistent. It is indeed complex because there are a lot of components involved, lot of interactions involved, lot of what to say basically elements involved, be it different kinds, say genes, transcription factors, proteins, etc., etc., etc. So, like, yeah, these are complex, and obviously it is not homogeneous in nature. Next question. This is a single answer question, by the way. Please keep in mind while working on it. Okay. So the question is, which of the following is true regarding Michaelis-Menten kinetics? It gives the final rate of product formation for a given substrate concentration is absolutely not true. It is an example of a stochastic model. No, you cannot infer any kind of statistical information from Michaelis-Menten kinetics. A low value of Km indicates the binding of the enzyme with the substrate is high. So yeah, a low value of Km corresponds to a low value of Vmax by 2. So basically, your the reaction speed is increased basically with the binding of the enzyme and the substrate because it is very high. So the low value of Km will indicate that the binding of the enzyme in the substrate is very high. Um, the Vmax for for a reaction remains unchanged on doubling the enzyme concentration, which is not true. The Vmax will definitely change. If the enzyme concentration increases, the Vmax will reach, will slightly increase and then reach a plateau, because obviously there is competition for the enzymes. But yeah, it will not remain constant. And the equation is unchanged when inhibitors are present. So no, this is not actually true. And uh, I am not remembering. I don't know if I remember correctly. I think in Leninger's book also you'll find there are various different, uh, basically different methods, not methods, basically various different uh, complexities to the Michaelis-Menten kinetics. So you can have it when activators are present, you can have it when inhibitors are present, how the Tmax and the Km value will change when these kind of elements are present within the reaction. There are there are mathematical equations which you can derive on how your reaction rate changes with the presence of inhibitors or activators, that can be derived using mathematical equations. And I think it is in Leninger, if I'm not very wrong. And if I'm wrong, just Google it. You'll find it. There are mathematical equations that you can derive to prove that Vmax is unchanged. Vmax changes when inhibitors or activators are present inside the system. Okay, moving on. Next question. Okay, so before I move on to the rest of the questions, I know the whole course. In the course, you have MATLAB lab lab videos and coding videos, but I will not particularly, meaning when I post coding questions like this, I will not particularly post it using like MATLAB syntax. These are essentially pseudocode syntaxes, so I'll be following pseudocode syntaxes only. And one thing for um, these MCQs, and for this particular lecture only, the basically the indexing is starting from one. So it is not in this question. We'll face it in the next. I think the question after this or after that. There are questions where uh, I have used indexes of arrays or lists to iterate. And just to keep in mind that the index starts from one. Generally, obviously it starts from zero. When we are indexing, when we are writing codes, we index from zero only. But this is a little bit different. Just to give you a heads up, it starts from one. So yeah, I'll give you 2 minutes. Okay. So if you look at the loop, basically it'll loop for when I equal to 3 and when I equal to 4. So it'll loop for two times. It doesn't matter on the value of p, because the value of I doesn't depend on the value of p. So the number of iterations in the loop will not be dependent on the value of p, instead on the number of values I take. So I will take two values, and so the number of iterations in this loop will be two. Moving on to the next question. I'll give you 2 minutes. This is also a single answer correct question. So okay. So the question is, if an enzyme catalyzed reaction, so you have E plus S giving an ES intermediate, which gives you E and P. Basically, P is the product, S is the substrate, and E is the enzyme. Is proceeding at near or maximum reaction rate of Vmax, what can be deduced about the relative concentrations of S and ES? Now, E is something that is in the reaction. E is an enzyme, so it is not getting used up in the reaction. So it is forming that intermediate, and then it is breaking apart to form get the product, and you're getting the enzyme back. So enzyme is not the limiting factor here. The limiting factor here is the substrate. So if your reaction has to reach the maximum rate of Vmax, the substrate needs to be higher, and ES will obviously be its highest point. So the answer will be that S is abundant because E is getting regenerated altogether. So you do not need to worry about E. So S needs to be abundant if you need to reach the maximum rate of Vmax, and ES will obviously be its highest point. So, yeah. Moving on, next question. I'll give you a minute or a minute and a half. This is a fairly easier question. Okay, so just to give you a heads up, these are basically the arrows that have like basically these, consider these are network with three edges and three nodes. So the three nodes are given by the ci, laci, and theta. Basically these are microbial networks. So anyway, so these symbols that like the edges that have that T kind of shape at the end, these are these represent repression basically, or down regulation. So theta is down regulating ci on the other hand is down regulating laci, and laci is on the other hand down regulating theta. So all the nodes in the network are down regulating each other, or repressing each other. So hence this is known as a repressilator. So it's fairly easy. Moving on to the next question. So, before you delve into the question, I'm sure a lot of people here are of biology background, but if you're studying systems biology, I'll suggest read up a little bit. If you do not have prior idea, read up a little bit about what is the rank of a matrix, what is the space of a matrix, what is the basis of a matrix, these kind of basic matrix algebra definitions, and try to understand a little bit about how these work, because these you'll need these going forward in the course as well, and also if you pursue systems biology in your career or in your research, whatever, you'll you'll need mathematical operations on matrices specifically. So I'll suggest to give it a little bit of read, not much detail. Like you don't need to know everything about matrices. Just you should know the basics. So rank is a basic. Like you should know rank. If you know matrices, you should know the rank of what is the rank of matrix. So yeah, I'll give you 2 minutes to solve this question. Okay. So basically, I'll define what a rank of a matrix is. Basically, here this matrix has three columns. So if you consider a vector space, it will have basically three dimensions. So one is sorry, four dimensions, because the number of rows in this matrix is four, but the number of columns is three. So you will have the first vector will be 1 0 0 0, next vector will be 0 1 0 0. So basically first vector will be I cap, next vector will be j cap, and the third vector will be k cap plus two of whatever you the fourth dimension like letter you imagine. So let us say n. So the third vector will be k cap plus 2 n cap. So how do you define the rank of a matrix? The rank of a matrix is basically the number of linearly independent columns of a matrix. So in this matrix are the all the three columns independent? Yes. Why? Because you cannot make one column by linearly combining these two. These two meaning the other two. So you cannot make the middle column by linearly combining the first and the third. Similarly, you cannot make the third column by combining the first and the second. Similarly, you cannot make the first column by combining the second and the third. And the term linear combination is very important. You need to combine linearly. You cannot do square root or stuff like that. You have to do linear uh, operations on this. So linearly, all three uh, columns of the matrix are linearly independent, and hence the rank of the matrix is 3. So moving on to the next question. Okay, this is a fairly easier question. So the rules of matrix multiplication is that the number of columns in the first matrix that you are multiplying should be equal to the number of rows in the second matrix. So if both A and B are of dimensions 3 by 2, you cannot multiply A and B because A will be 3 by 2. It has two columns and three rows. B on the other hand will have the same three rows and two columns. But to multiply, you need two rows, not three rows. So A and B would not be able to multiply. A and A transpose. Now A will be 3 by 2. A transpose will be 2 by 3. So if you multiply, you have the number of columns from the first matrix equal to the number of rows of the second matrix. So if you multiply, you will have a 3 by 3 matrix. Similar is the option of A into B transpose. So B transpose will obviously be a 2 by 3 matrix. A into B transpose will also give you a 3 by 3 matrix. Similar is the case of B and B transpose. So just keep this in mind that the number of columns of the first matrix should be equal to the number of rows of the second matrix in order to multiply them. Okay. So moving on to the next. Yeah, for this, just as I was saying, just remember that the indexing starts from 1, not from 0. So B of 1 will be 3, not uh, B of 0 is not equal to 3, B of 1 equal to 3. I'll give you a minute. This is also very easy if you are familiar with this. Okay. So basically, what it is doing is it is running a loop from 1 to 4, and it is summing up the elements. So basically, you need to sum up the first four elements. 3 plus 8 equal to 11, plus 9 equal to 20, plus 4 equal to 24. So the loop is running for 4 times, and each time it is summing up all the elements. So basically, it will be the sum of the first four elements, which is 24. It is a fairly easier one. Now, moving on. Not this one. Yeah, this one. This one is a little bit tricky. Just to give you a heads up, the value of b is 2a squared plus 3a plus 4 if it is not clear from how it is written. And yeah, I'll give you 2 to 3 minutes time because this might require a little bit more time for you. Okay, so let's look at the solution. Basically, you need to, as I said, the expression of b is 2a squared plus 3a plus 4. So when you calculate b1, it is actually the value of b according like the value of b corresponding to the first value of a. So b1 is the value of b corresponding to the first value of a. Similarly, b2 is the value of b corresponding to the second, and similarly the third. So if you calculate b1 will be when a equal to 2. So that expression becomes 2a square becomes 2 into 4 8, 3a becomes 3 into 2 6, and plus 4 becomes total 8 plus 6 plus 4 equal to 18. Next, when you come to b2, b2 you calculate 2a square, 2 into 3 square is 9, into 2 into 3 square is basically 18, 2 into 9 18, then 3a is 9 plus 4, that becomes 18 plus 9 plus 4 equal to 31. And if you calculate b3, it will be 2a square equal to, I mean, this in this case a equal to 5, so 2a square is 50, 3 is 15 plus 4 69. So if you add them up, 69 plus 31 plus 18 gives you 118. So yeah, this was the last question of the session. So I'll stop the recording here, and yeah, I think 2 hours has passed for us, and see you next time. And if you need anything, like if you want to, like if you have any doubts or anything, please uh, keep keep a note of the things, and you can ask me in the next session, next Tuesday 6 to 8, same time. And yeah, please attend these sessions and solve these problems, because these are the kind of problems you get in your assignments. This is for your own benefit, so please do join the sessions and do contribute. uh, so thank you everyone. See you next time.